So this is something new we haven't done before. Hello to everyone in the room and everyone who's joining us on Zoom. Thanks so much for joining us. I'm Tom Calvanese. I work for Oregon State University here as the station manager for the Port Orford Field Station, which is where we are and where you are beaming into. And this year things are a little bit different, but I'm excited that we did have a fantastic group of students this summer, and we can't wait to hear what they have to share with you. We have a team of students that studied gray whales here, joined by
studying whales, remember?
Darling, I can hear you.
Oh.
Yeah, so if you're tuning in for now, if you could mute your microphone, that would be great. That'll help the students focus on their presentations. And yeah, where was I? So we also have a Marine Studies Initiative intern who was studying the Redfish Rocks Marine Reserve, and he's going to share some of his experience there, where we quite literally immersed him in the Marine Reserve. And then he'll be followed by some students who were studying gray whales. And I can't wait to share this with you all and all of you here in the room. So without further ado, Tom McCambridge, please take it away.
And yes, all right. Hi, everyone. My name is Tom McCambridge. I am an incoming senior at Oregon State University studying marine biology. And this summer, I had the awesome privilege of doing an internship through the Marine Studies Initiative here at the Port Orford Field Station. And it was all centered around this question of interpreting the Redfish Rocks Marine Reserve. Slide, please. This here's the reserve. Ah, perfect. So the internship is basically centered around two central problems. So the first problem is with science in general and science communication. And the fact that science is kind of hard to reach for a general audience, and it's not really designed to be consumed by the average person. It's a little bit too disconnected from with what people actually tend to use resources for. And also the other problem is with the reserve. So there isn't quite enough exposure for Port Orford in general and the Redfish Rocks Marine Reserve. And I just kind of used this example of the Instagram pages. So the Monterey Bay Aquarium Instagram, they have, the Research Institute specifically has 60,000 followers. So people care about what's going on in the ocean. It's just a question of getting those people the resources and showing them what we're doing. And even when compared to a very comparable institution at Hatfield, that's also an OSU field station, they have a way wider range and a more engagement. But this is an awesome place, and we're doing awesome stuff. So people should be able to see it. And so that was part of my goal, is to get more people engaged with what we're doing. And so a central kind of mantra for us is to interpret the reserve, you need to have discrete experiences within the reserve to then show people and to use that information in the content that we put out. And so one of the major things that I was part of, you'll hear a lot more about it, so I won't go too deep into it, but is the GEMM Lab whale research team. So I went through their kayak training and their CPR training, so I was able to be kind of a substitute in their research process. So I was able to go out kayaking, as you can see down there with Noah and Liz. And then also, when you use equipment in the ocean, a lot of the time, it gets stuck in the ocean. So the other thing that I would do is kind of help dive down and get stuff off the bottom, which was a very unexpected but very cool part of the job. Slide. And there was a lot of emphasis on local fisheries, because Port Orford is such a fishing town, and there's so much dependence on the fisheries. And that's interesting when you look at what a marine reserve is. It's literally a place where you cannot fish. So there's a lot of interplay between what the fishermen think, what the scientists think, and then honestly, there's a lot of overlap there, because there's a lot of common ground. Like, everyone wants this to be a sustainable business. They want sustainable fisheries. And there was a lot of great experiences just in terms of marine biology, processing fish, like learning the parts of the fish to then fillet the fish, sell the fish. So Tian, the owner of Port Orford Sustainable Seafoods, the company that shares the field station space. Actually taught me how to process fish. I learned a lot about the local fish species through the fish selling process, just seeing what's around here. And so, yeah, the fisheries is a huge part of what makes Port Orford Port Orford. And there's also an emphasis on aquaculture, specifically in terms of urchins. Down at the port, they have an urchin kind of ranch, and they're farming dulse and then fattening up urchins. And this all connects to the kelp, because there's a big problem with too many urchins kind of booming, eating all the kelp, and then affecting the kelp forest habitat. So this is just another interesting kind of interdisciplinary approach to that problem by creating a taste for urchins. So that was an interesting learning experience. And also there was the intertidal research through PISCO, which stands for the Partnership for Interdisciplinary Studies of Coastal Oceans. Yeah, and they come down to Port Orford because there's like amazing intertidal rocky habitat. And so they have a bunch of long-term research processes, including measuring mussel beds, measuring predation on mussels by sea stars, and long-term disturbance projects. So they'll scrape an area clean and then over time study what replaces what was there. And so it's really interesting to learn where they go, why they go there, and what they're doing. So that was an awesome opportunity. And then having said that, there was also the community engagement aspect, was mostly through the Intertidal. So we organized this BioBlitz, where basically we tried to encourage people to download this app on their phone, where they can, it's called iNaturalist, and you can go and take a picture of an animal and it'll identify that animal, or do its best to identify that animal, basically. And that was a great way to kind of get people outside and engage people in Port Orford with science and get people involved, but also, adhere to COVID restrictions and not get a bunch of people together for some event. And there's also a pH sensor out at Rocky Point that I would help deploy. That wasn't a huge part, but it's interesting to see that that's going on here as well. Slide. So the real meat of this was figuring out how do you reach people without actually talking to people, because I was supposed to be at the visitor center, which is now closed, unfortunately, because of COVID. And so how do you replace that face-to-face interaction, those surveys, those guides that you would, and tours that you would guide for people? And the answer was basically through online content and through Instagram and through YouTube. So the main social media outlet that I focused on was Instagram, because there's so much beautiful visual things about this area that I think people would love to see, and Instagram is a great way to get that to them. And so by using relevant hashtags and the Instagram algorithm magic or whatever, we were able to grow from 214 to 276 followers, which is a 25% increase in followers. And it's also like active people. Like, it's not just like random people just click follow and never look. It's like we're getting more likes, more comments. So it feels good that people are like seeing what we're doing and interested in all that. And then YouTube is the other main way. I've spent a lot of time making YouTube videos, and we went from 2 subscribers to 12 subscribers, which is a 500% increase. And so, yeah, like speaking of YouTube videos, you hit the slide. No, it's the like intro. Yeah, perfect. The first video and only video out now, there's another one specifically about the whale folks, so stay tuned for that. The first one is about what makes redfish rocks so special. And so just a little context, we're gonna play the video here for you, so you'll be able to, that's a lot more impactful than what I would say about the video. But yeah, just keep in mind, this is synthesizing everything I've talked about and trying to put it in a compact, succinct, 4-minute YouTube video. And so a lot went into it. I tried to really focus on the story of how redfish rocks become so abundant with life and give a good highlight for the redfish of redfish rocks. So that being said, the video is about 4 minutes. So pop some popcorn, get comfy, and yeah, I think that's all for me for now. Yes.
This is the Redfish Rocks Marine Reserve. On the southern coast of Oregon, just south of the small town of Port Orford, you can find this small 3-square-mile reserve and attached marine protected area. But what makes this area so special? Well, it's actually a whole bunch of things, and it all starts with the rocks. The large rock formations provide structure for many animals and plants to attach to and live inside of. In the spring, strong winds cause nutrients from the bottom of the ocean to be driven up towards the surface in a process called seasonal upwelling. These nutrients act like a fertilizer in the shallow water. These nutrients are used by phytoplankton, which spawn in large volumes and provide the base of the food web. The nutrients are also used by many types of marine algae that grow on the rocks. Probably the most important of these is bulk kelp, the world's fastest growing plant. When conditions are right, they can grow up to 10 inches or 25 centimeters in a single day. Bulk kelp basically acts as the trees of an underwater forest. Feeding on the phytoplankton, you can find zooplankton, which are actually many different types of animals. They can be mysid shrimp, amphipods, larvae of larger animals, or many other species. These zooplankton provide food for tons of different animals, ranging from tiny crabs nestled in the rocks, all the way up to the giant gray whales that migrate through nearshore waters in the summer. Many animals attach themselves to the rocks, including the bright green and blue anemones. These animals stick to the rocks and catch prey such as crabs, zooplankton, or fish with their stinging tentacles. Large colonies of mussels also attach to the rocks and filter out algae from the seawater. Colorful sea stars can be found moving slowly along the rocks using their tube feet and eating the mussels and other attached creatures. In the water, small fish use rocks and kelp to hide from the larger predators in the open ocean. They can form large schools like these smelt. Of course, there are fish that eat these fish. One example is the lingcod, which lays on the bottom and ambushes fish as they pass by. And what would redfish rocks be without the redfish? Many species of rockfish can be found here, and many of them are bright red, like the vermilion rockfish. Some species of rockfish can live to be up to 150 years old. Because they live so long, they need to be protected so that they can get old enough to reproduce before they are fished. The old female fish are especially important because they produce exponentially more offspring each year. This is only a fraction of the life that can be found in these waters, not to mention the land animals that depend on marine life. Many species of birds feed on life in the ocean. Brown pelicans, seagulls, and black oyster catchers are just a few of the birds that can be found on these beaches. Altogether, these creatures create an incredible mosaic of life that needs to be protected, and that's just what the Redfish Rocks Marine Reserve is doing. Thanks for watching.
Thank you. So yeah, I hope you enjoyed the video. I hope you like that. Hopefully you learned something. That would be ideal. But yeah, just real quick, I want to thank the Marine Studies Initiative and Port Orford Field Station. Of course, Tom Calvinese, my mentor through all this. and GEMM Lab, specifically big love for GEMM Lab, and Pisco. None of them are here, but thank them in spirit. But yeah, that's all from me. So I mentioned the gray whale and GEMM Lab research a little bit, but now to tell you much more about that, what they've been doing and what they've been finding, I'd like to introduce Lisa Hildebrand and the rest of the GEMM Lab team.
Hello, room. Hi, Zoom. I hope you can all hear us again. Yes, welcome to our annual community presentation about our gray whales here in Port Orford. Before I introduce our team, I also just want to highlight that this is our 6th consecutive field season that we've been here in Port Orford, so we're building up whoops in the room. We're building up a really sizable data set here and are so excited to keep being able to come back to this town and this community and to start working more with locals in lots of different ways. And so before I introduce the people on the team, I want to explain briefly our team name. This year we are called the Theodolers. And we actually can't take any credit for this name. We have to give credit to Scott Holt, who is the father of Matea, this year's OSU undergraduate intern. And at the beginning of our field season, he asked, Scott asked Matea whether the theodolite, which is the instrument we use to track whales from shore, is anything to do with yodeling. And it isn't, but we thought it was hilarious. Well, except for Matea, she didn't think it was so funny. But the rest of us overruled her and decided that it was going to be our team name. So without further ado, here are the Theodolers. And so I'm Lisa Hildebrand. I'm a master's student in the Department of Fisheries and Wildlife at Oregon State University. And this is the 3rd year that I am leading this project. It was first established by my advisor, Dr. Lee Torres, who you'll hear from later, who is an associate professor in the department, and her first master's student, Florence Sullivan, back in 2015. So, like I said, we're lucky enough to be in year 6 of this field season, and it would not have been possible without my 3 other Theodolers, who you will hear from in a little bit, but just so you can prepare yourselves from who you'll be listening to. First up, we have Noah, who's a fellow graduate student. He is currently completing a degree in marine science and management at the Free University in Brussels in Belgium, but he is a native Portlandian. Woo, Oregon. Then we have Matea, who is going into her senior year at Oregon State University this fall. She is our OSU undergraduate intern this year. And last, but certainly not least, we have Elizabeth Kelly, who's going into her senior year at Pacific High School here in Port Orford. So you'll hear from Noah, Matea, and Liz and myself again over the next half hour or so. But to take it away first, we have Matea, who's going to talk to you all about gray whales and a little bit of background about the whales that we study here in Port Orford. Yeah, so the gray whales that we study here are part of the eastern North Pacific population. There are about 27,000 of these whales, and they're baleen whales. So what that means is they have these plates of keratin, the same stuff that our nails and hair are made of, and they use them to filter feed on zooplankton. These whales migrate every year from their winter breeding grounds in Mexico to their summer feeding grounds in the Arctic. And they're extremely agile for baleen whales. Although they can be an average of 12 to 15 meters long, they can feed in water that's as shallow as 1 to 2 meters, as you can see in this video of a whale feeding by the port. So more specifically, the whales we study here are part of the Pacific Coast Feeding Group. What this means is that they feed here in the Pacific Northwest instead of going all the way up to the Arctic. This is about 200 to 300 whales, and you can see on the map that the blue dotted line shows the typical migration up to the Arctic, and the green line shows where the Pacific Coast Feeding Group stays in the summer. These whales are defined as any whale seen from Northern California to Southern British Columbia after June 1st. But really, for them to be considered a true part of the group, we want to see them multiple years. And a big part of this project is kind of asking, why do some of the whales stay here while the rest of them go up to the Arctic? Now, one key difference between this sub-feeding group and the population as a whole is that they feed in slightly different ways. So up in the Arctic, they primarily feed benthycally, which means that they suck up the sediment into their mouths and filter out the zooplankton there, and that consists primarily of amphipods that you can see on the right. And they also do that here, but in the Pacific Northwest, there's another way they feed that'll be in the water column, and when they're feeding there, they get amphipods and also mysis, as you can see on the left. Now, what whales are we seeing here? Well, so we actually take photos of them to ID, and we can do this because they have unique pigmentation on their dorsal hump and their fluke. And this is cool because it allows us to kind of follow whales through the years, through different places, and see kind of what they're doing, where they're going year after year. And we also don't see all of the whales across the entire range. Here are a few of the whales that we see in Port Orford. This one is Smudge. We first saw Smudge in Port Orford in 2019, and he's named after this circled white mark that looks like a smudge. And Smudge is the most commonly seen whale for the last 2 seasons. This is Swoosh, first seen in Port Orford in 2018. Swoosh is named for the dark mark that's circled on the left, and he is the second most commonly seen whale during this field season. Now this is Basil and her calf Twig. She was first seen in Port Orford in 2018, and she has a very distinctive large propeller scar on her left side. We saw her this year, and South Coast Tours also did, and got this picture of her in the bottom right corner. And I'm sure a lot of you have heard about Button. We've seen him every year in Port Orford since this project began in 2015, and he's named for the white dots at the base of his fluke. And now that I've told you a bit about these whales and what we're doing here, Noah's going to tell you how we study them on a day-to-day basis.
Okay, hello everyone. What's it like a day at the office for the gray whale team? Nothing like this poor whale here, but nonetheless exciting. So we start the day around 6 a.m. Those of us who are coffee drinkers have our cup of coffee and enjoy the view from our field station that is unbeatable. After the cup of joe, we head down to the lab, and the kayak team and the cliff team prepare our equipment for the day. On the left, you'll see the equipment that the kayak team puts together, and on the right is what the cliff team brings up to Fort Point, and I'll explain all those pieces later on, but those are our daily tools. Around 6:25, the cliff team heads up to Fort Point with the theodolite, the camera, and our laptop, and those are used for tracking the whales, like physically tracking the whales. The laptop lets us save that data so we can actually use it. And for the camera, we try and take pictures of the whales so that later on we can photo ID. Around 6:45 a.m.-ish, the kayak team, after getting set up with their wetsuits down at the port, launches at the port of Port Orford, which you can see, this is our launch site, and heads out for sampling on the water. We tend to try and go to Mill Rocks. We have 2 sampling zones. So we have the Mill Rocks sampling zone, and then we have another sampling zone sort of behind the port. And we tend to go to Mill Rocks first to try to evade the common afternoon Port Orford winds, and then head to Tichenor, try and get as many as we can. Around 7, we usually arrive at our first sample station. whether it's in mill rock practitioner, depends on the day. And it takes, well, it says 2.5 hours. I was being nice, probably more like 3.5 hours to get our sampling done for the day. And that's if we get a full day, so barring any weather or equipment issues. But what exactly do we do at each station? So the first thing we do is we drop a tool called a Secchi disc. And you can see on the left the video playing. We drop the Secchi disc into the water column, and as soon as the white portions are no longer visible, we stop dropping it. And as you can see, Liz is pulling, well, she's dropping it now, but then she'll pull it back up, and there are little notches on the string, which she counts as she pulls it up. And each notch accounts for 0.5 meter in distance, and that's the visibility of the water column that day. The second tool we drop is the GoPro camera and the TDR, which helps us measure the depth at any given point throughout the drop. The GoPro footage is used to get a relative quantity of zooplankton in the water column. And sometimes the GoPro footage is cool, like this, fish, lots of zooplankton, although not in this one. And sometimes the visibility is so bad that the GoPro footage is hard to see anything. And last but not least, we drop the zooplankton net. And the zooplankton net is what we use to get a representative sample of the abundance and diversity of the zooplankton that's at each sampling site. So Liz will attach this net, and then she'll drop it in the water. And it has a weight on the bottom, and the net will drop all the way to the bottom of the water column. There she goes. And as soon as it hits the bottom, she'll pull it up as hard as she can, as fast as she can, to try and catch as much from the water column as possible. It's a good arm workout. Yeah. There she goes, working it up, gets the zooplankton, pulls it up, and then once she has it up in the boat, she uses a little squirt bottle and tries to get as much of the, as many organisms out of the net as possible into a Tupperware sample that's labeled for each site, and then we throw that in the bin and go to the next station, do it all over again. So now Lisa's going to take over for a minute.
I'm just going to jump back in, and you might be asking yourselves, so you've sampled for prey at 12 stations, now what? So Noah's just talked you through the two ways that we sample for the prey in the water column. We have these GoPro drops where we get videos, which we use to assess quantity of prey. So how much prey is out there. And then the net samples, which you just saw Liz really haul up from the bottom of the ocean, is what we use to assess the quality of prey. So what kind of prey is there? And the way those two methods work is for each GoPro video that we have, we take still images every 5 seconds. So this is an example of one of those still images. We then divide it in a 3x3 grid, and we assign each of these boxes a number from 0 to 5. 0 meaning there was absolutely no prey, 5 meaning, oh, there's a lot of prey. And so just as an example, I've worked through this still image here. I've given each box a number. Once that is completed, I take the average of this number to get just one number for the whole still image. Once we complete that for all the still images of that drop, we sum all those numbers together, and that gives us a relative quantity of the prey that was there. So we'll be able to say, wow, that station had a lot of prey, or there was hardly any prey at that station. And so how we then translate this further is, I'll talk you through a little example. We're just going to zoom in on mill rocks for this example. And so what we have here are basically the numbers of that method that I just talked you through. And as you can see, there's that one station in the center that's red and has a bigger number than all the others. Most of the other stations are 0 or very low, and then we have that one station at the top that's kind of in the middle. And so while this is great, it means that we only know what is happening at those 6 precise locations. But I want to know, how much prey is here, or how much prey is down here. Because as much as I shout at the whales to go to this specific location, they never listen to me. They're going all over the place. They're going all in this gray area where I have no idea what the quantity of prey is. And so a way that we've been able to kind of try and predict or estimate what is in those gray unknown areas is by using these values that we do know and using the habitat type and the distance to kelp to try and figure out what's happening in these unknown gray spots. Because we're finding that this zooplankton tends to be closer to kelp and on more rocky habitats rather than on sandy bottoms. And so by using those 3 things, I'm able to create these kind of visual maps that are predicting what's happening in all those gray areas. And so what you're seeing here is wherever it's bright green, that's where we have high levels of prey, so lots of prey. And anything that's kind of pink and going into the white is indicative of low amounts of prey. So I'm able to do this for every day that we go out and sample and we get these GoPro videos to create these kind of maps of what's going on in each of our study sites. Then I also discussed quality of prey, so what kind of prey, and this is linked back to those net samples. So once we bring those up and we go back to the lab, we actually painstakingly, for hours, most of the time my interns, thank you Theodolers, sit at a microscope and look at every single individual prey item that we find and try to identify it to the species level. And the reason we do this is because we're trying to see whether these gray whales have preferences for different kinds of prey. And so just as an example, what you're seeing on this graph, on the horizontal axis, you have different stations. So from 1 to 19 are different stations. And on the vertical axis, we have just the percentage of the prey community, so of the net sample, made up of different species. So as you can see, station 1 is 100% in this teal color, which is one type of mysid shrimp. And station 8 is 100% of this brown color, which is a different species of mysid shrimp. And so you might be wondering, well, why does this matter? What does it matter if there's different species? And I'm going to tell you. So this next plot is, you are one of the first people to see these results. This is primarily what is going into my first chapter for my master's. But basically what I did is, once we sorted out all these different species of prey, I ran them through a machine called a bomb calorimeter, which sounds very intense, and it kind of is. But we use it to figure out the caloric content of whatever you put into the machine. So I wanted to see whether these different prey species that we have out here on our Oregon coast differ in terms of calories. So kind of like if you eat a burger or a salad for dinner, that's going to mean different things for a human. And so I'm wondering whether burgers and salads exist in the gray whale world. And as you can maybe see, is that it seems like there are. So our biggest box over here, that is Dungeness crab larvae, and those seem to really be the gold mine. That's what you want to go for if you're a gray whale, because it's very high in calories. Versus something like an amphipod, which, as Matea told us, are more benthic dwelling, and they really aren't that much of a gold mine. They are pretty low in calories. And so this is why we really do this very fine scale identification of prey, because there clearly are differences and potentially would be benefits of only feeding in areas where you can, hit lots of crab larvae, or maybe hit some of these mysid shrimp, which have higher calories than these amphipods, which seem to not be very beneficial. And so with that, I'm going to hand it back to Noah.
So Lisa and I have kind of explained the method and the reasoning for the kayak sampling. But while the kayak team's out on the water taking samples, the cliff team is taking samples of their own using the theodolites and the camera. You can see a picture of Matea on the left, and that's her using the theodolite, which is the tool that we actually use to track the whales as they move through the water. But you might be asking, gray whale team, what is a theodolite? A theodolite is a tool that we, like I said, that we use to track the whale as it moves through the water, and it gives us a precise location of the whale as it moves. Really basic description. We set up the theodolite at the same position every single day, and we know the elevation that the theodolite is at, and then using another reference point to the side or behind it, usually land-based also, that theodolite can then calculate exactly where the whale is moving through the water, which gives us track lines, which I'll show you in a second. And on the bottom right corner here, this is a cartoon version of what it looks like to see a whale through the theodolite. And we'd be up on the cliff, and one of us would see a spout, and they'd say, blow. And then the other person would move the theodolite until the scope looked exactly like that, with the whale reaching. That person would say, fix. The person would click some buttons on the computer, and that's one data point. Lisa is going to explain to you more about how she's going to use the track lines, but this is what a track line looks like of a whale as it's moving through our observation.
Move on to this, I think we may have someone whose audio is on online, so I just ask that you mute yourself so we don't get any feedback from other microphones. So yeah, Noah just explained to you how we track whales from shore, and again, you might be asking, so you track the whale, now what? Well, the goal of this very fine scale kind of research that we do here is to bring together those two data streams. The data that we collect on the prey, which I just talked about, quantity and quality, and then to match it up to the gray whales, to really understand the kind of dynamics between these two groups and to see whether these individuals have those preferences for, you know, do they just go for a lot of food or do they really care about what they're eating? And so the way that I'm going to do this is going to be the next couple months of my life before I defend my master's in December, hopefully, is I'm going to talk you through a track line of Smudge, who Matea introduced to all of you before, with his beautiful white fluke right here. And so what you're seeing is our study site. We have, you know, Titchener Cove over here with our 6 sampling stations there. And Mill Rocks is over here, kind of obscured by all of these dots. And so each of these black dots represents a point where Smudge came up to the surface to breathe. Our clip team saw that and fixed Smudge's location with the theodolite and the laptop. And then these dots are connected by this black line, which just connects dots in a chronological order. So you can kind of see Smudge travels in a very unidirectional way. Kind of looks like he knows exactly where he wants to go. And then it gets kind of messy. There's a lot of dots in here, you know, big cluster of dots. And so to try and understand why we're seeing these cluster of dots, especially in this tight area, that's when we start to bring in this prey data. So I'm going to overlay this prey map that I walked you through earlier. And then if we bring back Smudge's track line, we can see that actually this big cluster of dots is happening mostly in that green area. So that area where there's lots of prey. Whereas in these areas where it's more white or pinkish, it's more of that unidirectional travel. So kind of Smudge moving through and wanting to get to the good stuff. So potentially, Smudge might only care about lots of prey. Might just go where it's green, where there's lots to eat it all, and then leave. But potentially, those findings may change as I spend the next couple months working through these 6 years of data, which is really exciting. But that's kind of what I'm going to be spending my next few months doing. And with that, I will bring it back to Noah.
Okay, back again. So yeah, Lisa's shown me a bunch of pretty pictures, how she uses all this data to explain what's happening here. But at the end of the day, when the kayak team comes back and the cliff team comes off the hill because of wind, because it's just the end of the day, we have to take all of that data that we've gotten, all of the organisms that we picked up in the water, and process them. So we have one team that focuses on freezing the zooplankton so that it doesn't squirm when we're IVing it. and then thawing it and using the microscope on the left to ID each individual organism. Yeah, there you go. Okay. Yeah, okay. That's good. Yeah. That's a highlight. Anyway, so then we get the abundance and diversity of the organisms at each station. Then we also have the data from the clip team and from the GoPro and the TDR, and we use the laptop to organize that data, and then Lisa can take it. So we have an ideal that we want each day to be like, and the reality is that field work never happens exactly how we want it to. Each day brings different challenges, whether it's weather or equipment or people not feeling well or whatever it is. And a big portion of our jobs is to focus on trying to adapt and overcome those things and still pick up as much data as we can. So Liz is going to tell you a little bit about what we've observed so far this year.
Alrighty, let me just get set up really quick.
Okay, so you guys have learned a little bit about what we're studying, the prey that we're studying, and the manner in which we're doing it. So I'm going to give you guys a little more as to how the field season has actually gone. So Secchi depths that Noah mentioned is how we measure the depth of the water, the visibility. In the beginning of the season, for the first 4 weeks, we could see further into the water. It was deeper, meaning the water was probably more settled, it was clear. However, in the last 2 weeks, our Secchi depths have become shallower and shallower, meaning there's more activity in the water, it's murkier, less visibility. And for zooplankton, the prey of our gray whales, we had very few in all of our samples throughout all of our stations for the first 4 weeks. And just over 2 weeks ago, we had a large, fast, rapid climb in zooplankton all over. And for the star of the show, the gray whales, for the first 4 weeks, we saw a couple. I think our count got to like 11. And then we would see them start to come in large numbers. And when we were seeing them in the first 4 weeks, we saw them out by the Redfish Marine Reserve or over by Humbug or over by Hell's Gate, which is not in our field of study. So the question there is, why were we lacking in whale sightings? Well, when there's a lack of predators, usually there's going to be a lack of prey, and this is totally true for our situation. Because we were lacking in zooplankton so much, we weren't seeing any whales really. And however, once the zooplankton sightings started to climb, so did the whale sightings. Why were we lacking in zooplankton? Well, actually, the nitrates at the bottom of the ocean are what feeds the phytoplankton, which is what feeds the zooplankton, which is what feeds the whales. And those get stirred up so that they're brought to the surface of the ocean so that they can grow and reproduce and be able to provide each other with the nutrients that's necessary to be an abundant food source for the gray whales. Which leads us to upwelling, which is what stirs it all up. I'm going to explain to you what it is and why it matters. Upwelling is this really important occurrence in which the coastal winds will go across the surface of the water and move the surface layer offshore, where the lower water, which is cooler with more nutrients in it, rotates to the top onto our shores, which is then what cultivates and grows and reproduces and becomes an abundant food source for our whales. So what is the cause of the lack of whales and zooplankton? First, this graph is going to help me explain that, but I'm going to explain it to you. Here on the x-axis, the horizontal, we have
from December of 2009 up until currently. And then on the y-axis, the vertical one, we have CUTI, which stands for Coastal Upwelling Transport Index. Here at the red dotted line, that is when upwelling and downwelling are completely neutral, meaning that there's no upwelling, there's no downwelling, neutral. And then this dark blue line that you see, it'll go up and down around that line. And when it's below the line, that means that we are having downwelling, which is just a reversal process of upwelling. And when it is above that red line, that just means that we're having upwelling. And now you'll see typically here, and what we are going to have green little boxes, you can see those are typically in June, which is the peak. However, the upwelling, as you can see, usually begins in about May, or the start of like summer. It'll build up, and that's when we're supposed to have our peak upwelling, which is when the ocean is nice, it's mixed, and it's ready for some whales. However, as you can see in these red boxes, that is not always what happens. Sometimes when we are supposed to have a peak, we have a dip, which is what we had this season, which has caused delayed upwelling. But now, as you're going to see here in just 1 second, we have a peak that is now occurring, which is why we're just now getting a huge influx of zooplankton and whales as in the last 2 weeks. Here is a video from the first, I think it's the 2nd week of our field season, where you can see the fish a few meters away. So the ocean is more settled. You don't see many, if any, zooplankton. Not a lot going on. So no upwelling. And then here is a video from about a week and a half ago that we took. There is an abundance of zooplankton. It's murky water. You can't really see more than a meter or two in front of you, meaning peak upwelling. That's all. So before we kind of wrap up here, now that the theodolers have told you about their season, more of, the science part, what we do on a day-to-day basis, I wanted to give each one of the theodolers an opportunity to share with you all one of their most memorable experiences of the summer, something that really stuck with them. And I'm just going to kick us off. So Tom very, very briefly mentioned this, but I want to give kind of the theodolers side to this story. So obviously we're working in a very rocky habitat, and we're dropping these weird shaped GoPros and nets into this very rocky habitat that has holes and crevices and tight spaces. So if you're working in an environment like that, it's really not conducive to you putting things in there that aren't meant to be there. And so every time we drop something off of our kayak, we know that there's this possibility that it could get stuck. But we're always, especially me, who's now in my 3rd year of doing this project, I'm always very confident that, if I just tug the GoPro in the right way, and if I don't let it down the bottom for too long, that won't happen to me, and that I'm always going to be okay. And so up until this year, I had never gotten it stuck, and I was always very proud of myself, until one day, pretty early in the season, I think it was week 2, Liz and I were out on the kayak, very first station of the day. We were feeling good. It was a calm day, flat waters, paddle out, and I dropped the GoPro, and I'm about to reel it up, and I feel that it's kind of stuck. And I was like, no worries, I'm gonna give it some slack, I'm gonna slowly try and reel it in. 2 hours later, we're still sitting there, and the GoPro is still stuck. So to my heartbreak, we have to cut the line, we have to leave it in the water, and try and figure out a way to kind of get this GoPro back. Thankfully, we have Tom and Tom, who are super adventurous, love to try new things, and were immediately down to help us and try and dive for it and try and get it. Unfortunately, at that point, it was kind of already almost midday, so the wind had picked up. So we agreed that we would wait until the following day in the morning when it would be a bit calmer and at low tide, so that Tom and Tom wouldn't have to dive so far to go get it. All great, no worries. Also, because we know that we can lose gear in this rocky habitat, we have backups. And so next day, it's all good. Matei and I go out. I was feeling that my ego was a little bruised because I lost the GoPro. So I felt the need to prove to myself and everyone else that I could sample just fine and that everything would go okay. So we paddle out, we do more rocks, we skip 19 because I do feel a little iffy about it. We finish at Mill Rocks, we paddle over to Titchener Cove. At this point, Tom and Tom are slowly getting ready on land, suited up, getting ready to head out with the assistance of Dave and his beautiful new boat, the Black Pearl. And it's while I'm at station 8 that I'm dropping the GoPro, and oh, that telltale sign that it's stuck. And once again, an hour and a half later, I can't get the GoPro unstuck. Thankfully, Tom and Tom were currently in Mill Rocks and had retrieved the first GoPro, which I was so excited to hear. And since they're already out on the water, they just speed across on the Black Pearl, make it back to Titchener Cove in no time, and they attempt to dive for the GoPro that I had just gotten stuck again. Didn't quite work that day. Thankfully, the next day on a Saturday, both of them were very willing to go out and try again. And they did get it back. And so thank you so much, Tom and Tom. You're the field team's heroes this year. Yes. But yeah, it was a big lesson for me to never feel too confident in my skills. It was very humbling, and I'm very grateful that we had Tom and Tom around to kind of pull that off. And with that memorable experience, I'm going to hand it off to Liz.
Okay, so I was in a kayak with Tom, and he was helping sub out for one of our other interns. We were out there paddling around over in Titchener Cove. We were sampling and stuff. Everything was fine. And I got a little radio call, and Lisa was all like, oh, hey, so there's a whale coming your way. Like, no big deal. Don't get in its way. Keep doing samples. I'll tell you when to, like, stop and back away. So I'm thinking to myself, it's fine. Everything's fine. And so this whale is just, like, coming our way, and Tom and I are like, Lisa will tell us when to move. And I'm thinking to myself, oh, that whale is super cool. And like throughout the season, I'm making jokes about like riding whales and taming a pet whale. Wow, okay. And then we're out there and we're sampling, and the whale just keeps like getting a little like too close. And I'm like radioing Lisa every once in a while, and I'm like, hey, yeah, the whale's like right here, and we're backing away from it. We're trying to give it like 50 meters of space. We're trying to stay away from the whale because we can't get too close. We want to give them their space. And so Tom and I are finishing a sample. We put it back up, and I say to Tom, hey, Tom, we need to back away, like just get offshore a little bit, give the whale a little bit more room, because I don't really know where it is right now. It's been pretty unpredictable with its surfacing. So he's like, okay, cool. And we're like backing up, and we're getting away, and then I hear this giant, loud, terrifying sound, and I look over, and like just a few feet away from me, and I get like this huge blow right in my face from its nasty, stinky breath, and I feel like it just coughed on me or whatever. It isn't very COVID friendly, but whatever. And it's terrifying. And I thought the whole season, I was like, oh, I want to get so much closer to the whales. I want to see the whales so close. But all I did was pee myself and scream and wave my paddle. And I was just like, ah! I was literally just screaming. And Tom was just like, ah! And I was just panicking. And then I'm like, move. And we just start moving. It's like 4 times. I don't know what's up with this whale. Like, they're non-aggressive. And it just won't stop chasing us. So I'm like in the kayak with Tom, and I'm like literally like going as hard as I can, and I'm kayaking, and I'm scared. And this whale is like trying to like rob me or something, and it won't stop like chasing us. Like I remember repeatedly, it would like line up with us, and I'd like see its spine like go directly down the center of the kayak, like 10 meters in front of us, and I'd be like, the other way then. And it just kept going. But it was also really cool. Because I was scared, but this whale was also like really close. So I guess it was awesome to be coughed on by a whale.
So something that I've enjoyed throughout the entire season is just going out in the early morning, kayaking, especially when the water's calm. Just nice way to start the day. And I really enjoyed kind of being able to see all the different wildlife that'll come up. And one morning I was out with Lisa, and we were just going. I was navigating, so I was kind of paying attention to where we were headed. And Lisa goes, "Did you see that?" I was like, "What?" It's like, "Oh, there's a hovercrabs over there." And so I just looked over, surfaced again, just one time, but I saw it. I was like, "Okay, cool. That's, you know, perk of field work to get to see cool other animals sometimes." And then we continue going throughout the season, and every once in a while we see cool stuff. And I kind of, you know, thought that, oh, we see more stuff on home days, and it's just, you know, when it's calm, it's good, and good conditions mean good everything. And then one day we were out, I was with Liz, and it was super windy. One of the windier days that I was out on. We weren't really having the best of times. This was actually, we were being blown around by the wind, trying really hard to get to our next station, and it was just the most difficult part of paddling that day. I was thinking, this is not a good day of sampling. And then we were struggling and looked over, and there's a harbor porpoise. And this time it surfaced maybe 10, 15 times. So we really got a good look at it. And that kind of gave me a little bit more motivation to go, oh, okay, you know, even if the conditions aren't perfect, it can still be a good day of sampling. And so we got to the rest of the stations, finished the day, and that was a nice little reminder.
Okay, cool. I promise this story is G-rated. The most memorable experience for me this summer was all of us coming from really different backgrounds, scientifically, culturally, life experience, and we all just like clicked immediately, became friends, communicated really well, and at the end of every day, we'd all sit outside at the tables, outside of our station, and we just talk and hang out. So we did the science during the day, and we were friends at night, and that was really special and unique. And that's it for me, really.
And so to wrap up for today, Lee is going to come up and just say a couple words. Yeah, take it away, Lee. Thank you. Hi, yes, hi everybody. I'm Lee Torres. I am the behind the scenes person on this project. So I want to introduce myself and congratulate the team on another awesome year. So as we said, this is our 6th year of this project, and every year it's different, but every year it's awesome. And I really am impressed with you guys. You know, I know you come in here 6 weeks ago, not really knowing what we're gonna give you, but you guys performed great. And I know there are bumps along the roads, but this is the secret of this project. The bumps are supposed to be there. The bumps are what the learning moments are, and that's where you gain your skills and you learn about teamwork and you learn about perseverance, and you all have done that this season. So congratulations, you passed, and hopefully you take from this experience, you know, new lessons that'll lead you further down your career, wherever you go in life. So, yeah, so great work. So we actually intentionally added another bump to this field work. So as Lisa and the team explained to you, what we're doing here is trying to understand the very fine scale association between where whales are and where their prey is. But we decided to take it even one level finer and go underwater and try and film the prey, the mysid, the zooplankton that you've heard so much about, and the whales. See if we can actually capture some footage of a whale feeding on mysid. So that's a big ask. So this year we called it the pilot season. But what we're interested in knowing is how the zooplankton aggregate and move and how they come together. And then what happens? How does a whale actually feed on it? How does it open its mouth? Does it suck it in? There's so many basics that we still don't know about how whales feed here off the Oregon coast. So this is our pilot season. And here I'm going to introduce to you CAM-DO. So what this is in this black cylindrical object there is a housing, wait, go back, is a housing for a GoPro camera. And within it has extra batteries, and it basically allows us to keep it at the bottom of the ocean for a long time and run. And then these are actually the brake pads from my husband's old truck attached to the lander. So this is the lander that he made for us. And our dive team, who was supported mainly by Aaron Galloway from the Oregon Institute of Marine Biology, the University of Oregon, and his team, and Dave Lacey, and that's his beautiful black pearl there. So we're so grateful for the support of these various groups that really helped us achieve this pilot project this year. So anyway, you can see the dive team putting our gizmo underwater, and we really didn't know what we'd find, if it would film, what we would get, anything. So you here are privileged, and actually I'll just say before we go forward, this thing is still in the water. So right by the jetty. So at first we had it by Mill Rock, but as you do in science, you have to adapt with what you find, and so we actually moved it to Titchener Cove, where we saw the whales feeding more and we were catching more zooplankton. So we moved it, and just this last week, hot off the press, we managed to actually capture some footage. So the first clip you'll see here on the left, you'll see just a bunch of mysids. You know, these are these clouds of zooplankton that we're so interested in. And you can see the rockfish down at the bottom, which also feed on these mysids, these zooplankton. And so we're interested in how they move and aggregate and what causes dense aggregations versus not. So, wow, you know, Lisa and I were like just pretty thrilled to get this. Okay, it filmed, it worked, we got some interesting stuff. Now here's the money shot though. Well, not the money shot, but there it was. That was it, a marine mammal. See if you can play it again. It goes by so quickly. But I mean, that's pretty thrilling for us. So it's still out there. We haven't gone through all the footage from last week's deployment, but just seeing this, woo! Pretty awesome. So we're really excited about this new component that we did have to work through logistically. I want to thank a lot of people. So that's what I have here, my list of people to thank. So specifically about the CAM-DO work, I want to thank Kevin Book and Taylor Eaton from the OSU Scientific Diving and Boat Safety Program that really helped us figure out how to get this thing in the water safely. Aaron Galloway and his whole dive team, obviously they were huge collaborators on this project and really made it happen. And Dave Lacey and South Coast Tours and your boat. I mean, we couldn't get out there to have our team in the water without you guys. So big props to all of you. And Tom Calvinese in the back there, he's just the man behind the machine here. So, and then we're going to talk about the whole support for this whole program. So again, we're in our 6th year, and every year we sort of add in different people that help us make this happen. So it is a real village that makes this project go ahead. So in terms of funding, I want to give a big shout out to my home institution, the OSU Marine Mammal Institute. So they were really the big funders behind the project this year. And the funding came from the license plate fund. So if you've seen the gray whale license plate, Oregon gray whale license plate, and if you have one on the tail of your car, you supported this project this summer. So I want to give a big thanks to everybody that bought those license plates. And if you don't have it yet, you can get one still. They're beautiful. And the money basically goes to the Marine Mammal Institute to support programs just like this, to do research and outreach and education. So we encourage you to keep renewing that license plate or buy it if you don't, because that's what makes this project happen. We also got co-funding from the Wild Rivers Coast Alliance. And I'll tell you, they've funded this project every year. So we're really grateful for the continued support of this project and really, you know, have made a whole cohort of interns succeed and move on. And then, of course, we also get funding from Plum, Level, and Square, who fund the Port Orford Field Station Foundation, and they, of course, provide us with a lot of support. Now, personally, there's also a lot of people that just give a lot of in-kind support. So obviously we got Dave Lacey and South Coast Tours, which again stepped up and they give us wetsuits and kayaks and training and just all sorts of help. So grateful. Tom McCambridge, who you've already heard from, he helped us out a lot this year. We're super thankful. I want to thank Minda Stiles and Mark Wilkie. They're the Marine Mammal Institute backbone that help us with a lot of logistics to make it all happen. I want to thank Barney Arita, who has allowed us access to our cliff site, which is obviously very important so we can track our whales. I want to thank Oregon Sea Grant, who also gives a lot of support. I told you there are a lot of people that support this project, but I got to name them all. And then obviously I also want to thank all the families, the people that support the interns, you know, that allow them to come here and for 6 weeks and dedicate all their time here instead of doing whatever else they do with their lives. So thank you for the families. Two more very important people to thank. Tom Calvanese. He really is a partner, the man behind the whole field station, and he's a huge supporter from day one of this project. We were just talking about how 6 years ago I met him and I was like, I want to study whales here. And he was like, okay, let's make it happen. And here we are 6 years later, you know, really a good legacy that's only going higher and forward. And lastly, I want to thank Lisa. She is an amazing leader, which you all know. But she has really persevered, grown. It's been such a pleasure to watch her grow into this role, to learn, to see her learn, and the data that she's collecting and going to be analyzing for her master's is just really educating us so much about the lives of gray whales and how they feed and live here along the Oregon coast. So good job, Lisa. Okay, with that, I'm done, but it's time for questions. So from you all, as well as online, for the students and Lisa. So please take it away. You wanna see that? I don't know. So stop sharing.
Turn their radios on and ask their questions.
Oh, hello everyone. Let's see. I see there's some things in the chat. Oh, well, this might actually be a question for Tom Calvanese. We have, what keeps the abundant fish population seen in the videos alive during the periods when the upwelling and productivity are low?
Oh, I didn't realize I was going to get pulled in. Question hour. Well, the thing about fish, and particularly a lot of rockfish species, are opportunistic predators, and they eat what's out there. So when they don't have access to one food source, they'll switch over to another food source. At 1 month, it might be juvenile crabs that are coming around that they eat. It might be baby squid. It might actually be other baby fish. They actually will eat the young of their own species. They're just, like I said, opportunistic. So they kind of go with the flow and they eat what's available. So when we don't have the upwelling, we don't have the plankton out there for them to eat, they'll find something, trust me. Hopefully that answers the question.
Eric's iPhone, what species was that on the camera footage? If you're talking about the footage at the very end, that was a harbor seal. That was the marine mammal that we saw swim through the camera. How many residents do we have in Port Orford? Any of my interns want to answer that question? Oh, Robert Ivy, do you mean whales or do you mean humans? I guess we should clarify that. In the meantime, does anyone have a question they want to ask? Yep, that's what I was referring to. Thanks. Which one, Eric? Eric was talking. Oh, sorry, Eric was talking about the CamDo footage. Does anyone have a question they want to ask in person if they want to unmute themselves, or just type another question? Oh, okay, Robert Ivy, whales, of course. Does anyone of my interns want to take this? No? Okay. No takers. I'll take it. So I don't know if what we have of the whales that we see here are necessarily just Port Orford residents. Definitely Buttons is one of those whales that we've seen since 2015 every single year. But as Matea said when she gave that kind of background about gray whales, is that we see, for the most part, these Pacific Coast Feeding Group whales move kind of through the entire range from southern British Columbia to northern California. So we think, we're pretty sure that we see whales have kind of a preference for certain areas, but I don't think we can say that we have just Port Orford residents. In our photo ID catalog, though, just for Port Orford, in the 6 years that we've been doing this season, we have about 30 whales. So that's kind of the different kinds of whales that we've seen here specifically, but they don't just go here. Nathan, if you want to unmute yourself and ask your question, go ahead.
I want to introduce you.
Oh, yeah. Nathan, can you introduce yourself?
Oh, hey, my name's Nathan. I'm actually an alumni with the GEMM Labs research group. I was an intern in 2016, I think. And then 2017, yeah, that sounds about right. And I also helped sub in for kayaking a year later. And my question is actually about the data visualizations you guys did. I thought it was really cool how you took the prey density map that you created and the whale track lines and how you were able to show how they correlated. And I was curious if you were thinking of doing something similar with the caloric content data of the different zooplankton species. What kind of visualizations are you thinking of doing with that?
Yeah, that's an awesome question, Nathan. So to be honest with you, for this presentation, that visualization, I really just pasted that track line on top. I knew that they overlapped in terms of the coordinates, but in the haste of the field season, that's kind of just what I did for this presentation. But you're totally right. One of the plan is to then kind of, I guess it would be multiplying that kind of percentage of what species we see at the different stations by the caloric value to kind of get a calorie map of what is in the environment. I haven't worked through that yet, but that is the plan.
Yeah. That's super cool.
Yeah. Good to see you, Nathan.
Good to see you too.
Paul McCambridge, have you seen any changes in the amount of prey over the years? Does any of my interns want to take this anecdotally from what I've told you? Liz shrugged. Is that a yes?
Sure.
Yes. Liz, take it away.
From what I've gathered about essentially just listening to Lisa,
Which that might be wrong. No.
Potentially. This year we've seen definitely a change in the zooplankton because of our delayed upwelling that I spoke about. So there was definitely way fewer, not only whales, but zooplankton as well. But I think throughout the years that there's like pretty natural dips. This isn't the first time we've seen the delayed upwelling, and I assume that we'd also have a lower count in zooplankton and whales when there is the delayed upwelling like there was in 2016 and 2012.
Yeah, exactly.
I could be wrong, but
no, that was perfect. Yeah, good job, Liz.
Yeah.
And definitely also what we see is we see this kind of, as Liz said, these peaks and these dips as the season goes on. So the way I kind of see that in my head is when we have, a bunch of whales, they'll come in and kind of graze down that prey. Then we'll have a couple days of low prey, time for the prey to kind of reproduce, increase their abundances. That's when we kind of don't see any whales. And then when that prey kind of comes back up, we see whales move back into the area. So there is definitely these natural ebbs and flows that we see. But with the delayed upwelling this year, that's definitely something where we had a very big chunk of our season with very low prey. Thanks for the question. Anyone else?
Yeah, anybody in the room? Yeah.
Oh.
This is like a splinter group of gray whales. What the estimated total population of gray whales?
So for anyone who didn't hear the question was, what is the kind of total estimated number of the whole population from which this kind of Pacific Coast Feeding Group is a subgroup, right? And so that main population, the Eastern North Pacific population, is about 27,000 individuals. So it's really big. So this subgroup is a really small portion of that population. And so it's kind of fascinating that only the small number is deciding to cut the migration short by almost, a half, more than a half.
which is why, you know, having 6 years of data start to build up, we can hopefully start to address some of those questions. Okay, Maria Hildebrand. Oh, that is my sister. Hi, Maria. Great distinction about calorie high and calorie low items. Is there also plans to research whether whales need more protein, fats in their diet? Is it possible to see it like that? Ooh, Lee.
What was the end of that? Sorry.
Is it possible to see it, like like distinctions between protein, fats, things like that? I guess the way.
In the zooplankton?
Or I guess in terms of what whales need. I mean, I guess that is, yeah.
Yeah.
You wanna?
No, you can
Okay.
take it to the behavior level. I think the behavior and the BAI, is that what you're talking about?
I don't think so.
You take it.
Okay. Well, if the question is whether the prey can be broken down into kind of protein, fats, carbohydrates, that kind of thing, then that is something that can be done. It's kind of lipid analysis to see how fatty different prey species are, and that has been done for other species. We currently don't plan on doing that, but maybe in the future. So yes, there's always a potential to do more with the prey. Tara Ramsey, is Port Orford the only location that you study? One of my interns knows the question to this. Anyone? The answer to this. Mateo, Noah, who wants to take it?
Sure.
Noah, yay.
Yeah, so for this particular group of gray whales, Port Orford is the only place that we studied this particular zooplankton content, but there's also gray whale studies coming out of Newport, studying some different dynamics of the gray whale population.
Yeah, that's fair. Yeah. So the manner in which we study them here in Port Orford with the fine scale cliff tracking and kayaking is pretty unique just to hear. Lee does run a different gray whale project out of Newport where we fly drones and we collect fecal samples to to assess changes in body condition of gray whales. So we can assess that from drone footage to see how fat or how skinny whales are across over the season. And we collect fecal samples to assess their hormone levels to try and assess whether they're in nutritional stress or other kinds of stress and things like that. Thanks for the question, Tara.
Yeah, another here.
Oh, yeah.
What are the caloric rock stars of the North Pacific? Are they more calorie rich than the foods that we have here?
Good question. So Kian asked what the caloric rock stars are of the Pacific Northwest in terms of prey, and compared to the Arctic, how do they do with the calories? And so Kian, that is exactly the question of my first master's chapter. Very nice. And so definitely not quite, not quite there yet on the chapter, but yeah. So definitely for the Pacific Northwest, Dungeness crab larvae is kind of the rock star. That's the one that the whales should try and go for, but of course those are seasonal, so they're not as constant as kind of the zooplankton. So the mysid shrimp are also seasonal. We tend to only see them mostly in the spring and summer, but obviously they're not as cyclical as Dungeness crab. And so a whale feeding here, a gray whale feeding here, couldn't just survive on crab larvae because it's not always here, whereas the mysid shrimp are almost always here in the summer. Compared to the Arctic, I'm not quite finished with that part yet, but it seems like so in the Arctic, there seems to be less of a variation of prey that they eat. They kind of only feed on 1 or 2 species of these amphipods, which are those things that bury in the sediment. And we're actually finding that they're pretty comparable in their calories compared to the mysid shrimp here, which kind of adds this interesting component, because if they're comparable, then why wouldn't more whales come here? Like, why not cut your migration short by a half rather than go up to the Arctic? So it's probably a question of just density, so how much prey a whale can get in like a mouthful of food. My guess is that up in the Arctic, it's much denser. They can get so much more of those little amphipods in one bite than the mysids that we see here. Yeah, pretty comparable. Steve Garrett, what other whales are you seeing in the area? Oh,
Liz can answer this question for sure because she and I saw another species of whale.
Yes, so just like, I don't know, like 2 or 3 days ago, Lisa and I were up on the cliff with the theodolite and whatnot, and we were like just up there and we saw a few blows. Well, Lisa saw them and I thought she was lying because she said they were massive. And I was like, okay, massive blows. And they were in fact like very massive. And then we went over and it was too far for us to track the theodolite. It was out of our zone of study. But she did manage to get a couple all right little pictures and she sent them over to Lee Torres, the leader. And she identified those as 2 blue whales. So the really big whales.
Yeah, largest animals on the planet. So, and they really weren't that far offshore. Steve, I don't know if you're familiar with the Port Orford area, but they passed right behind Hell's Gate. So I don't know how many kilometers offshore that is. Not many. 2, maybe 2 or 3. So they were really close in, which was pretty cool. Yeah, but we also have, humpback whales probably use this area offshore. I know a lot of the fishermen have seen sperm whales a couple miles offshore before. So yeah, definitely lots of other things that use the area too. Any other questions in the room? Any final questions online before we say goodbye?
What are your plans for next year?
Oh, okay. Tom Calvanese, our awesome station manager, asked what our plans are for next year, and they are to come back. So if you are a high school student a local high school student like Liz, big footsteps to fill there. She's really been awesome. Or an OSU undergrad, or any kind of other students like Noah, grad student, and you're interested in kayaking, seeing whales, just learning about science. Keep your eyes peeled for an announcement for next year, because as we've shown that even in a pandemic, we can make it work, and we will persevere and continue to study these awesome whales. So thank you, everyone. Oh, yeah, I will pass it on to our station manager, Tom Calvanese.
No, I'm good. I just want to leave you with one final message. Thank you, everybody. Give these people a huge another round of applause. Just amazing work, and in this context, to be able to actually do good science and do it safely and with respect to everyone, I think was just a huge accomplishment. I'm so grateful to Lee and your whole team. You're just total rock stars, in my opinion. And I can't wait for next year. And I want to give one last plug, which is if you like what you see and what you hear, you can also support this station and the work that's done here and the students and people who come here to work. Go to the Port Orford Field Station website and you can find a place where you can make a donation. And if you can't make a donation, just send us good vibes, because we're going to keep at it and we're going to get through this okay. And I'm really looking forward to doing this again next year. So thanks again. Thanks everybody for tuning in, and thanks everybody for being here.
Look at all the great comments we got. Thank you. You were spot on with your last question.
Great job. You can get it.
Awesome. Good work. Great work. Liz, you were awesome. I'm really impressed. That was a complex topic to tackle. You
fights over.
Oh, you guys are so well, so proud of you. Hold it off.
Yeah.
What were your tasks?
Liz, yours?
Yeah, so, yeah, we look like we're letting it hilarious.
Hilarious.
So,
Yes.
I know, the whale, their breath does people are always like, oh, you have whale, oh, it's so disgusting. I love that. Oh, yeah. Smell, and then what I say they're.
But if you're dying, it's like, you're not living.